Global warming is the cause of a rise in temperature all over the globe, which has been shown to increase the growth rate of photosynthetic organisms. Raven and Geider, 1988. One such photosynthetic organism is algae. It can be a very important part of the ecosystem because it provides food and refuges for small organisms. However, if the growth rate of algae increases, there is a strong possibility that we may see an overgrowth of algae. This can lead to algae using up dissolved oxygen that other organisms need to survive. There are also some algae, such as red algae, that produce a toxin which can be harmful to animals and humans. O'Neill and Limby, 1995. This can lead to an algal bloom, and more algae could create a hazardous environment for marine life and can affect life on land through contamination of resources and the possibility of toxins becoming airborne. U.S. Department of Commerce, 2013. We hypothesize that as the water temperature increases, there will be a positive correlation with the metabolism of the algae, and this would lead to an increased growth rate. We decided to run our test with a type of filamentous algae known as Spirogyroglyphalia. This species has an optimal temperature of 22 degrees Celsius, Stefan 2015. We used two different incubators with two different temperatures, 22 degrees Celsius and 30 degrees Celsius, to test our hypothesis. The Spirogyroglyphalia samples are split between 4 flasks filled with a standardized growth medium along with 2 flasks without algae to act as controls. We massed out between 0.09 and 0.11 grams of algae for our samples. The 2 control flasks were given no algae, only growth medium as well as some deionized water as a control for the experiment to make sure there was no contamination. Then we put 2 of the algae flasks and 1 control flask in the incubator with 22 degrees Celsius, and the others were placed in the incubator that is 30 degrees Celsius. Samples were left for a week and a half to grow. First, we gathered the wet mass of the algae samples and compared them to the starting mass. We attempted to measure chlorophyll A through spectroscopy as another way of calculating mass, but the results were abnormally low, indicating an error in the measurements. The method we used to measure dissolved oxygen was the Chemetric R7512 dissolved oxygen test. This test used vials that contained chemicals which changed color based upon the amount of dissolved oxygen. We used long tweezers to break the vacuum-sealed vials under the surface and pull some of the medium up before reacting. As the vials changed colors, we compared them to the pre-tested vials that read from 1 to 10 parts per million.
The dissolved oxygen in the control flasks at both 22 degrees and 30 degrees Celsius registered at 8 parts per million on the dissolved oxygen test. There was no trend between the two. The average dissolved oxygen level of samples in the 22-degree incubator was higher at 6.5 parts per million with a standard error of 0.5 than samples at 30 degrees Celsius at 5 parts per million with no standard error. This leads to a downward trend in dissolved oxygen levels as temperature rises. The starting mass for the algae set at 22 degrees Celsius and the one set at 30 degrees Celsius was between 0.09 and 0.11 grams. At the end of the experiment, the wet mass of the algae set at 22 degrees Celsius increased to an average of 0.71 grams with a standard error of 0.06. The flask set at 30 degrees Celsius increased to an average of 0.80 grams with a standard error of 0.06. This shows that there is an upward trend as temperature increases. The data that we collected indicates a relationship between water temperature and increased metabolic rates, which lead to increased growth. However, there is a need for more experimentation using other types of algae. These experiments are important because the growth rate of algae greatly affects the ecosystem that it is growing in. The effects of algal blooms often result in a decrease in dissolved oxygen. As dead algal matter descends towards the bottom of the lakes, it gets broken down by bacteria using dissolved oxygen through respiration. Christensen and Leeds, 2016. Large algal blooms can clog the water surface and create a thick layer of algae that could make it hard for organisms like turtles and frogs to breach the surface for air. O'Neill and Limby, 1995. Other algal species such as Rhodophyta can produce dangerous toxins, harmful to aquatic organisms in high quantities. These toxins could contaminate resources such as fisheries and have the potential of becoming airborne and harming human life. U.S. Department of Commerce, 2013.